PowerPoint 1 Part A. In this picture, I would like to point out date or times of bacteria. So if you look, it's approximately 3.2 to 3.5 billion years ago that it was first discovered. So this is the picture, similar to the beginning of your lecture outline. You again have right here the parent cell, which then undergoes replication and divides into the two daughter cells. This is happening between generations of cells. And within that particular cell, you also have gene expression, which includes transcription, making mRNA, and translation, making protein. And finally, and the second half of this entire unit is talking about between cells of the same generation when there is a recombination. You should be able to see there is a darker purple in this area of genetic material and a lighter one right here. So bacterial chromosomes or bacterial genetic material is just referred to as a chromosome, and it is in fact circular. But when you include that as well as the plasmid discussed in Unit 1, then together this is known as the bacterial genome. So again, the genome is a combination of all of the genetic material within the bacterial cell. So again, here we have the genetic material, which is right here. This is our nucleoid, if you think back to Unit 1. And right here, this little standalone piece of material is the plasmid, also seen on the picture on the right. And collectively, all of this DNA is known as the bacterial genome. So now we're going to take the time to go through some of the components of DNA replication. Up top is a replication fork, and this is what is traditionally known and recognized when you studied replication or DNA replication for eukaryotic cells. Below here, you can see a bacterial chromosome because it is circular. And this is going to be the process for prokaryotic cells, but once we go through the eukaryotic one, you'll see right away how the two are very related. At this point, if you do not remember all of the different enzymes and the components that make up this particular replication fork and how the process of replication works, go look into the supplemental module for Unit 2. There are multiple videos going through the steps, and you can watch them in a live animation. So a couple of terms that you need to be familiar with, and this is related to the fork up above, is that DNA replicates in a bidirectional fashion. Another term for that is that it is anti-parallel. So DNA grows in a 5' to 3' direction, but it actually reads the strand because of this bidirectional fashion in a 3' to 5' manner. The first thing you should see over here to the left is that DNA is the double helix and wrapped together, held together by bonds. So the first step absolutely has to be for this particular enzyme to come in. And this enzyme is the helicase. ACE implies that it's an enzyme. It will come in and unwind the DNA. And next, these little balls and enzymes attached, they are known as single-stranded binding proteins. These function to keep the strands separated because they need to be single-stranded in order for replication to occur. Just by looking at this picture, you should be able to recognize that the top particular strand is known as continuous, or also known as the leading strand, whereas the bottom one is discontinuous and also known as the lagging strand. If you think back to when you first learned this in biology, the continuous strand only has one primer, whereas the discontinuous strand has multiple primers. A primer is 5 to 10 nucleotides, and a primer is made of RNA, not DNA. So if we take a look first at the discontinuous strand down here, not only do we have a primer, we also have DNA polymerase 3, which has the job of polymerizing the DNA. So the two begin putting in the bases. Make sure you understand why the discontinuous and lagging strand have multiple primers, whereas the continuous strands actually have just one primer and one DNA polymerase III. If you look at directionality, because we did say that DNA grows in a 5' to 3' direction, on the continuous strand, this is the growth going in a 5' to 3' direction. And then again, because things are anti-parallel, that will tell you that the parental strand, as opposed to the daughter strands, begins at 5', 3', and ends at 5'. So if you take a look at the bottom, you have your RNA primer right here to the left. So this is growing in a 5' to 3' direction. So if this daughter strand is a 3', that implies that the parental strand is a 5' here and a 3' over here. Please look at directionality to make sure that this flows for you and that you understand how DNA is replicated. So once this process is done, in the continuous strand, it's okay because you simply have one single primer. But in the discontinuous strand, there's multiple primers. And this creates a problem because, again, a primer is made up of RNA nucleotides, and we want DNA. So then comes the job of DNA polymerase I, which comes in to essentially remove the RNA primers. And then the final enzyme, I need you to be familiar with ligase. If you ligate something, you glue it together. So once DNA polymerase I inputs the DNA to where the primers were once located, ligase will come in and glue the entire thing together. So that's DNA replication summarized for you. Now, if you take a look at the picture on the bottom and relate it to the top, notice on the top you have one area where replication began. This area is known as the origin of replication, and in short, it's known as ORI. If you take a look at the bottom, this is your ORI. Again, the area on the bacterial chromosome where replication is going to first begin. But please keep in mind that when the chromosome is circular, you actually have two ORIs because it unwinds itself and starts replicating in both directions. So once you have a comfortable understanding of DNA replication, we're going to move on a little bit to talk about the actual genetic map of E. coli. Notice how it is in fact underlined in this particular picture. E. coli has approximately 3,000 genes, and again, it's used often because it's well understood and its entire chromosome to a point is also mapped. First, you want to begin by understanding the difference between genotype and phenotype. So genotype is the actual gene, whereas phenotype is physical. It's the physical representation of what is or isn't synthesized. So if you take a look at this entire chromosome, notice the plasmid is here off to the side. So if we combine chromosome and plasmid together, this picture could be referred to as the bacterial genome because it's all of the genetic material. So let's take a look at this box right here. The E. coli has an allele for making tryptophan. This is the TRP. If I find another one, most of you are familiar with LAC, which is short for lactose, and there's a few other ones that you might be familiar with. Notice how it has trip written right here. So the genotype, does it have trip? Yes, genotypically it does in fact have the allele. But take a look next to it right here where you have trip and there is a very clear little minus sign. If that allele for trip were to become mutated, it would no longer have the ability to synthesize or make it. So if the question here was, what type of gene does E. coli have? You could in fact say that it has trip or tryptophan. But phenotypically, the answer is it does not synthesize it in this particular picture. So does not synthesize. And why? Because there is the minus. If you don't see anything, you assume it's a plus. It's only designated when a mutation has occurred and it can no longer make it. For example, genotypically, I can also say that the allele for lactose is present. Phenotypically, there is no minus signs, so this one does synthesize lactose. Genotype is if the gene is or isn't present, and phenotype is, again, its physicality. It does or does not synthesize it. We're going to have to talk a little bit about proteins and how they actually describe the phenotype of bacteria. Remember that enzymes are made of proteins, and we had talked about metabolism, which was anabolism and catabolism together. So these enzymes are necessary to carry out all chemical reactions. And then these enzymes actually characterize the bacteria. Most of this is a bit of review. We know that enzymes function to work with things such as cellular respiration, fermentation, all of the different metabolic processes. They make things such as adhesins, if you think back to pili, and endotoxins, if you think back to LPS. They also function as exotoxins, which we will be talking about in this unit. And then they work to help build all of the various cell structures the bacteria needs. And this includes things like the cell wall and the glycocalyx, also known as the capsule. So if this weren't in an educreations lecture, what would have happened in a classroom is this top list of genes would have shown up. And I would have said to the class, please count how many genes are actually in this list. And they all would have been black in color and looking like one overflowing sentence. When you are looking at genes, basically when there's a number, when there is a parenthesis, when there's a minus sign, that tends to separate the genes. So wondering how many genes there are, notice here is a minus sign. It is separated by color. This one right here might have been a little difficult. And then again, there's your parenthesis and a number and a number and another number. And finally, more numbers and a parenthesis. So if you add this up, there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. There are 14 genes. If you were doing this on an exam question, if your number was somewhere between 11 and 16, that would tell me that you understood lists of genes are written together, but the minus implies that phenotypically it is not synthesized, and a parenthesis is usually specific and also relates to the next gene in the list. So we've talked about replication. We've talked a little bit about what's happening at the point of division. And now we're going to look at gene expression, which is, again, a complete review. So on the next slide, I'm going to write out the enzymes that I need for you to be familiar with. And part of those videos for supplemental too, if you don't remember transcription and translation, I really need for you to take a look at them. And I'll mention one more time, recombination is going to be Unit 2, PowerPoint 2, and the second half of this particular unit. So if you're looking at this picture, the first thing that's important to know is when you take a look at the top or the bottom picture, how do they compare? And the answer is they're taken from two different textbooks and they are the exact same picture shown differently. If I say to you, is this replication or is this transcription? A couple of the things that you would notice is in replication, there was never notice of a tail growing off of it. For replication, you always have complementary base pairing. You have the adenine pairing with the thymine, the cytosine pairing with the guanine. This is that complementary base pairing and the bidirectional replication that DNA replication undergoes. In this particular picture, you should notice that there's also uracil. Uracil, when we're talking about RNA versus DNA, these bases are accurate for DNA. For RNA, it's a little bit different. Hopefully you remember this. Cytosine does still pair with guanine, but adenine tends to then pair with uracil, which is designated by the U. So once you take a look at a picture and you see uracil, or you see a tail sticking off of the end of what might look like a replication fork, that should essentially tell you that this is transcription. The purpose of transcription, or the end products that ends up getting made, is mRNA. If you take a look at the picture below, you're seeing the same thing. This enzyme right here is an RNA polymerase, because remember, for DNA replication, you had a DNA polymerase. ACE is an enzyme, and when you polymerize something, it's growing or it's synthesizing. Here, we're making a growing strand of RNA, or mRNA, because this part of the picture is representative of transcription. So then this is an RNA polymerase. And right here you have polypeptide chains growing off of the end. So these are proteins being made. So that picture is the start of translation. Final question is, what are these two steps to make proteins collectively called? Transcription working together with translation, and collectively together, this is known as gene expression. So here in the last picture, naming the process, there is your growing protein chain. So this process is translation, and you would know that because it is in fact making proteins. So the top picture as well as the bottom are again taken from just different books. So first you had the DNA and you replicate it. Then you transcribe that into mRNA. Then the final step is a translation of this mRNA, because this is your messenger RNA right here. And that is then translated into a growing protein chain, which is also known as a polypeptide chain. Some of the terms that you would need to be familiar with are, you want to know why AUG is important. And the answer is AUG is what's known as a start codon. A start codon has to be found in order for translation to begin. And then you have to assume that if there's a start codon, there is in fact also a stop codon. So this is AUG, and it codes for the amino acid methionine. This is the only amino acid that I suggest everybody know off the top of their head. Besides that, there are actually charts for amino acids, which is located for you on the Canvas page. So you want to understand how tRNA plays a part. This is your transfer RNA. They look like little dynamites on the top as well as on the bottom. They come in and complementarily base pair to the messenger RNA. If you take a look, here's your AUG, and the first protein is in fact the methionine that was coded for. So you want to know what a codon is, an anticodon, a tRNA, what's the significance of a start and a stop codon, and to finally watch the video of how once this polypeptide chain hits a stop codon, it's going to cleave or cut itself off for the sake of efficiency, and then the process itself will stop.